All right, we had progressed to this point in our lecture, still discussing the ideas that were at work during what we call the golden age of microbiology. Um, Now, on this slide, it mentions something that happened quite a bit before even Leeuwenhoek was around, and that is a scientist or doctor named Girolamo Francescoro in 1546 who had sort of a hypothesis that he called the germs of contagion. He probably wasn't using the word germ in the same way that we use today, although that word kind of stuck with it to some extent. But he was, at least he was saying there could be something, some particle that caused disease, not just bad air or some kind of curse on your father or some such. So the idea that there was like some kind of, physical particle, cell that could cause a disease was starting to be thought of. But that was 130 years before Leeuwenhoek even saw microbes under a microscope, so people didn't really understand what that would be at that point. But Pasteur, like I mentioned before, he was working to help winemakers figure out reasons for why their products might be spoiled or ruined, and really it led to what we call the germ theory of disease. Really, that microorganisms are responsible for contagious disease. There are other kinds of disease like inherited disease. That's not what we're talking about, but we're talking about the kinds of disease that come from the activity of microorganisms. Then you have this scientist or doctor named Robert Koch, and he studied the causative agents of disease, or what we call the etiology of disease. So etiology is a word that you want to know the meaning of. It just means the cause, the underlying cause of disease. And what Koch did, he was able to grow colonies of bacteria, mostly on solid surfaces. He used some kind of potato or gelatin material, and I don't have a real sense of what that looked like at that time. Today we use agar, so it was some kind of solid surface that he had come up with, but this picture shows an agar plate. This would be probably a blood agar, which blood has plenty of nutrients for organisms. And we will use some blood agar plates in the course. But anyway, in this example, they've spread a mixed culture of bacteria onto an agar plate, and each cell gets separated on the surface of this agar plate, and then grows by cell division, makes a big group of cells identical to it, and we call that a colony. And you can tell here that each colony, or that many of the colonies, have a different appearance than many of the other colonies, and that indicates different species. However, you do see some colonies that look similar to some others, and those might be the same species. So what Koch came up with, and you need to memorize, was the beginning of really applying logic to figuring out the etiology of a disease. Now, Koch's postulates work pretty well and was a very good start. It's not perfect in today's world, but it was the start of a very important series of events that helped really move microbiology forward. And so you do need to memorize these. So Koch said, okay, the causative agent, the microorganism, if it causes a certain disease, then it goes to reason that it should be found in every case of the disease, and it should not be found in healthy individuals. The agent has to be isolated from that individual and be able to be grown outside of the individual. And then when the agent is then introduced into a healthy person, that person must get the disease. And then you should be able to find that same microorganism in that second diseased person. So how they carried out all of these things, usually we wait until a natural inoculation happens. Whether or not they intentionally took someone healthy and inoculated them, that's hard to say. But the postulates allowed scientists and doctors to identify the cause of quite a few diseases, particularly protozoan caused diseases and really mainly bacterial diseases. This method mostly was not able to figure out viral diseases, although there is a virus that's on this list. But this list shows different scientists took Koch's concepts and were able to use them. And so very quickly you had many bacteria that were then associated with particular types of diseases. You should not memorize this list. Do not memorize. The concept, though, is that the work that Koch and Pasteur did then led to a whole bunch of scientists and doctors able to figure out with pretty good certainty what organism was causing what disease. And a lot of times they named the organism after themselves. So you'll notice that, like Albert Neisser calls it Neisseria gonorrhea, the bacterium, and so on. You see that several places. But hey, if you find a new bacterium, you're allowed to name it. Another really important scientist in this time is Hans Christian Gram. So the idea that we could find bacteria and identify it is, I mean, still a huge idea in microbiology. Bacteria do constitute a large amount of the pathogens that we'll be looking at. And what Hans Christian Gram did was he was able to figure out that bacteria fall into two main categories. And so if we're going to be identifying something, it helps if we can do some kind of test that immediately eliminates half of the candidates. And so what this technique is, it's called the Gram stain. And the Gram stain is the first stain you would use if you're trying to identify an unknown bacterium. In this photograph, in this micrograph, this is a light micrograph, we see a mixture of bacteria, and some of them are pink and some are purple, and that's because they've been subjected to the Gram stain. The ones that are pink are said to belong to the group of bacteria that are called Gram negative, and the ones that are purple are called Gram positive. And Gram negative and Gram positive bacteria, that's a major dividing line in bacteria. In other words, that's the first thing you want to know when you're trying to identify bacterium is it Gram positive or is it Gram negative? Because you would immediately be narrowing down the possibilities of the identity of that bacterium by half. So it's an important test that's done. Now, it doesn't identify the bacterium, but it does eliminate half of the possibilities. So we will learn how to do the Gram stain in lab. And just like there were a lot of doctors that figured out the etiology of many diseases based on what Koch and Pasteur did, we have another list that you do not need to memorize, but more scientists that were able to then take these ideas and put them into practice. In other words, if there are these invisible microbes that cause disease, what can we do in terms of medicine and nursing to improve the outcome for patients? And so what you start to see are these very pioneering types of work that were proposed by different scientists and doctors. And, particularly hand washing, aseptic technique, cleaning tools, sterilizing tools between patients, nursing in terms of washing hands and using and sterilizing things as you move from one patient to the next. So all of these basic prevention measures started really at this time as well. So you don't need to memorize this list, although I do think you may have heard of Florence Nightingale, who's shown in this picture. And she did a lot for improving the techniques that nurses use, but you do not need to memorize the list. So in the end, for this chapter, I did, in the first slide, I laid out some of the objectives, and one included a list of scientists that you need to know. So certainly, what did Leeuwenhoek do? What did Pasteur do? What did Koch do? And memorize those 4 postulates. What did Graham do? Those would be the 4. And then Aristotle, he was the one that we blame for the wrong concept of spontaneous generation. And in case you're not decided on your career choice, microbiology is a very good career choice. There's always a demand for microbiologists, clinical microbiologists, medical lab science. All of these things are fields of microbiology. And you can also get a job in microbiology in the public sector, in terms of like agriculture, public health, and then private companies, pharmaceutical companies, all kinds of things. So there are a lot of directions you can go if you really enjoy, particularly the practice that we're going to be learning in the laboratory. So if you haven't decided on a career yet, there's a lot of directions you can go from here that aren't directly into medicine, but maybe working in the laboratory at the hospital, for example. Or you could be the microbiologist at a food company to maintain quality control on their products. Or there's many, many places where microbiologists are in demand. So that's the end of the chapter 1, third part of the lecture, and the whole chapter 1 lecture at this point.